Hey, Bio2 students, Mr. Yance again. We're going to go over a little bit of DNA notes, the molecular basis of inheritance. We just got done with the genetics chapter and went over Punnett squares and Mendel's laws and theories. And basically, this is what later, after Mendel came up with these theories, we had evidence. We knew exactly how they work, and it's based on DNA. And you got to remember, Mendel. And back in that time period, we didn't know what DNA was. It came along much later. So this is basically proves all those Mendel laws that we talked about in that last chapter. Okay, so a little bit of history. The big question back in the 1950s was what causes these characteristics, what causes Punnett squares to work like that, what causes the law of segregation and independent assortment and those types of things. We didn't know if it was a protein or DNA. That was a big question in the 1950s. Which one was the genetic material? And in 1952, Alfred Hershey and Martha Chase performed experiments showing that DNA is the genetic material. They used a thing called a phage, which was called a T2. And what a T2 phage does is it invades and takes over an E. coli cell. So they set up this experiment. I'll show you that in just a second. And basically, by running this experiment, they proved that there is some type of genetic material, we call it DNA today, that is going to come along after the 1952. And it's not a protein. Okay, so it's a whole separate classification group, which that's what we're going to go into. So basically, at this experiment, they concluded that injecting DNA of a phage provides that genetic material. I know there'll be a little bit of an echo. Apologize for that, but let's watch this real quick. Come on. Just loaded this thing.
In the Hershey-Chase experiment, bacterial viruses called phage were used to demonstrate that DNA is the genetic material. The phage used in this experiment consisted of a DNA molecule surrounded by a protein coat. When phage infect bacteria, they attach to the surface of the bacterium and inject the DNA into the cell. The protein coat remains on the outside of the cell. In the first part of the experiment, phage were produced in a medium containing S35 radioactively labeled amino acids. This resulted in a phage population with S35 labeled proteins, but no radioactive label in the DNA. The phage were then allowed to infect the bacteria. The phage attached to the bacterial cell and injected their DNA, but the radioactively labeled protein coat remained on the outside of the cell. The phage produced in these cells contained no radioactivity. Vigorous shaking caused the empty protein coats to be removed, but did not interfere with production of new phage in the cell. In the second part of the experiment, phage were produced in a medium containing P32 labeled deoxyribonucleotides. This resulted in a phage population with P32 labeled DNA, but no radioactive label in the protein. When the phage infected the bacteria, the P32 labeled DNA entered the cell and could be found in phage subsequently produced in the infected bacteria. This demonstrated that the DNA, but not the protein, carries the genetic information for a new generation of phage.
Okay, so not a really, a little more complex experiment than you might think, but basically it proved that it's not just the protein and the DNA were two separate things in that phage. If they weren't, then they should have been able to see that radioactive material inside of E. coli and also inside the protein on the outside of the phage. So that's basically, that kind of started the ball rolling. So this is what really got scientists excited in the 1950s. They're like, oh, well, what is this DNA? And how does it work, and what's it look like, and all that good stuff. Because you got to remember, we're kind of in the dark at this point. Okay, now we know all this stuff today. but back then we didn't. So as soon as the biologists were convinced that DNA was a genetic material, then we had a big challenge. Let's determine, what's it look like? How does it work? How does it reproduce? How do we make blood, muscle, skin cells out of it? So along came Maurice Wilkins and Rosalind Franklin, and I really think I put Rosalind Franklin's picture on here because, she's the one that really should have been given credit for this. Wilkins was her professor or supervising professor and took credit for all of it. It was Rosalind Franklin that really came up with this X-ray crystallography and really started the ball rolling. She produced a picture, a pretty crude picture of something called, or of DNA. And you can see it here in the picture on the right. And that is what really gained momentum in DNA research. So once she produced this picture, okay, now we got a lot better technology today, but back then this was a big deal, okay? These two guys kind of piggybacked off of it, and I really wish they would have gave more credit to Franklin and some of them, but that's how science works sometimes. Watson and Crick, we've talked about them in Bio 1, okay? They came on the scene, okay, in 1953. So you can see how fast this progressed, right? They came up with this double helix model we still use today. You can see it back here in that picture I had up there before. They figured out that DNA was the substance of inheritance, and it's one of the most celebrated molecules of our time in biology. It is the, holy grail of how biological systems work. Without DNA, nothing living exists, right? And DNA is DNA in every living thing. I think I've told you that before in Bio 1. DNA is not different in a zebra or a fungus or us or an oak tree. It's just the sequencing that makes our diversity of life on this planet. So DNA is kind of an interesting thing. And remember, in 1953, Watson and Crick came up with this sophisticated, crazy, complex model, but they couldn't see it. We didn't see it until the 1960s, and they were spot on. So that was impressive, and they won the Nobel Prize. I just wish they would have gave some more credit to some of these other pioneers as well. But basically, we know hereditary information is encoded in DNA. It's reproduced in all cells of the body, regardless of what cell you want to talk about. It directs the development, the biochemical development, anatomical physiology, the function, the structure, the behavior to some extent. That about covers everything, right? We are our DNA. What we look like, how we function, all that is tied in with this most celebrated molecule of all time in biology, the DNA molecule, okay? And Watson and Crick's model had three parts. This should not be anything new to you, okay? It's a double helix. It can be described as a twisted ladder with rungs in it. The sides of it, if you guys remember, see if any of this rings a bell, is sugar and phosphate. And then the rungs of the ladder, the steps, are the four nitrogen bases. And I guarantee you remember these: adenine, guanine, cytosine, thymine. Should ring a bell. And if you guys remember, the A always goes with T, G always goes with C, okay? And what we found, and this is what I want to get in a little more detail with you guys, and I'm going to kind of wrap up notes today, and I'll explain this later, this 5' 3' thing. The outside strands are anti-parallel. Think of two trains running in opposite directions, okay? So one runs in the 5' to 3'. I'll explain this later. The other one goes from 3 to 5. So they're opposite anti-parallel strands is what you see. And we'll watch a video on this, and I'll put this online. You can watch it too. Might be a good e-learning thing someday. And we're going to talk about basically how do we replicate DNA, how do we transcribe it and translate it, and we're going to talk about those three processes and make it foldable and do all that stuff later. All right, so that's just a little bit of an introduction. I don't want to get into too much detail. Virtual students, I'll put some foldable instructions and color sheets online, and we're going to put it all together and make a little foldable out of it we can use on part of the test, and we'll start going through all this stuff, okay? But that's for another day. So, hey, have a good day. We'll talk to you later. We'll see you. Bye.